Hello everyone, my name's Ima and welcome back to my YouTube channel. Today we're going to tackle a practice problem set that relates to our lecture on lipid structure and function. Let's go ahead and get started with problem number 1. Problem number 1 says, which of the following is not a type of glycolipid? Glycolipids are lipids that have sugar head groups that are attached through glycosidic linkages. And on the MCAT, the most important glycolipids to know are Sphingolipids, which include cerebrosides, which have a single sugar, globusides, which have 2 or more sugars, and gangliosides, which have complex oligosaccharides and sialic acid. All 3 of these are definitely glycolipids, but sphingomyelin is different. Even though it's also a sphingolipid, it has a phosphate-containing head group, not a sugar. So that means it belongs to the phospholipid family, not the glycolipid family. So the correct answer here is D, sphingomyelin, because it's the only one that isn't a glycolipid. Let's go ahead and move to problem 2. Problem 2 says, during saponification, blank. A says, triacyl glycerols undergo a condensation reaction. B says, triacyl glycerols undergo ester hydrolysis. C says, fatty acid salts are produced using a strong acid, and D says fatty acid salts bind to albumin. Now, saponification is the process where triacyl glycerols are broken down in the presence of a strong base like sodium hydroxide. What's happening chemically is an ester hydrolysis reaction. The ester bonds between the glycerol backbone and the 3 fatty acids, they're cleaved, and they release glycerol and 3 fatty acid salts, which we know as soap. So with that being said, the correct answer is B. Triacyl glycerols undergo ester hydrolysis. Let's quickly review why the other choices are incorrect. Choice A says it undergoes a condensation reaction. Condensation forms ester bonds and releases water. But in saponification, we're breaking bonds, not forming them. So A is incorrect. C says the fatty acid salts are produced using a strong acid. That's incorrect. Saponification requires a strong base. Finally, D says fatty acid salts are binding to albumin. Now, this does happen in the bloodstream for transporting free fatty acids, but that's not part of the saponification process. So D is also incorrect. Once again, the answer here is B. Problem 3 says, which of the following describes the structure of steroids? All steroids share the same 4-ring fuse structure, and the key detail is in the specific composition of those rings. A steroid contains 3 cyclohexane rings, each with 6 carbon atoms, and 1 cyclopentane ring, which has 5 carbon atoms. This structure is what defines a steroid. And although different steroids may have different functional groups that are attached, the ring system always stays the same. So the correct answer here is going to be B, 3 cyclohexane rings, 1 cyclopentane ring. Let's also briefly rule out the other options. A says 3 cyclopentane rings, 1 cyclohexane ring. This has it flipped. C says 4 carbon rings differing in structure for each steroid. This is vague. It mentions 4 carbon rings, but it doesn't specify their identity. And it incorrectly suggests that the ring structure actually changes between steroids. In reality, the substituents vary, not the ring system. And finally, D says 3 cyclic carbon rings in a functional group. This is too broad and leaves out the 4th ring entirely. So again, the best answer here is B. Problem 4 says, soap bubbles form because fatty acid salts organize into a blank. Soaps are made of fatty acid salts, which have a hydrophobic tail and a hydrophilic head. And when you add them to water, they're going to spontaneously organize into spherical structures called micelles. In a micelle, the hydrophobic tails face inward away from the water, and the hydrophilic heads, they face outward towards the surrounding water. And this arrangement allows fatty substances like grease to be trapped in the center and then washed away. So the correct answer here is B, micelles. Problem 5 says, steroid hormones are steroids that blank. 1 says, have specific high affinity receptor. 2 says, travel in the bloodstream from the endocrine glands to distant sites. And 3 says, affect gene transcription by binding directly to DNA. Now that we've read through the statements once, let's try to figure out which ones are true and which ones are false, starting with statement 1. Have specific high affinity receptors. This is true. Steroid hormones, like all hormones, need to bind to specific receptors to exert their effects. And these receptors are often found inside the cell, either in the cytoplasm or nucleus, because steroid hormones are nonpolar and they can diffuse through the membrane. So 1 is true. What about 2? Travel in the bloodstream from endocrine glands to distant sites. This is also true. Steroid hormones are secreted by endocrine glands, like the adrenal cortex or gonads, and they travel through the bloodstream to target organs. This is what makes them hormones. They act at a distance. So 2 is true. What about 3? Affect gene transcription by binding directly to DNA. This one's actually misleading. Steroid hormones, they do not directly bind to DNA. Instead, they bind to intracellular receptors, and then this hormone receptor complex binds to DNA and regulates gene transcription. So while gene transcription is definitely affected, the hormone itself doesn't bind directly to DNA alone. And this is something we didn't explicitly cover in this chapter, but we will in chapter 6, where we really focus on DNA. So it's okay if you didn't know this one. It does require some information from chapter 6 of our MCAT biochemistry course. So statements 1 and 2 are fully accurate. That makes the correct answer for problem 5, C. Problem 6 says, why are triacylglycerols used in the human body for energy storage? A says they are highly hydrated and therefore can store lots of energy. This is actually incorrect. Triacylglycerols are actually hydrophobic, not hydrated. That's one of their key features. They store energy without binding water, which makes them much more compact than something like glycogen, which is heavily hydrated. So this statement is false. B says they always have short fatty acid chains for easy access by metabolic enzymes. This sounds tempting, but it is not accurate. Fatty acid chains, they can be either short, medium, or long, and triacylglycerols often contain long chain fatty acids, which are not necessarily easier to metabolize. So this is not the key reason. C says the carbon atoms of the fatty acid chains are highly reduced and therefore yield more energy upon oxidation. This is in fact the correct answer. Fatty acids have mostly single bonds between carbon and hydrogen, meaning the carbon atoms are in a reduced state. When you oxidize these carbons during metabolism, you release a lot of energy. That's why triacylglycerols are more energy dense than carbohydrates. They provide more ATP per carbon. Let's still work through D. D says polysaccharides, which would actually be a better energy storage form, would dissolve in the body. Now, this is misleading. Polysaccharides, like glycogen, are actually not more efficient for long-term energy storage. They're great for short bursts of energy, but they're hydrated and they're bulky, so they take up more space and they offer less energy per gram than fats. So this statement is not accurate. Once again, the correct answer for problem 6 is C. Let's move on to problem 7. Problem 7 says, which of the following is correct about fat-soluble vitamins? Statement 1 says vitamin E is important for calcium regulation. This is incorrect. Vitamin E's primary role is as a biological antioxidant. It protects cells from oxidative damage by neutralizing free radicals. It is not directly involved in calcium regulation. That's more the role of vitamin D. So this statement is false. Statement 2 says Vitamin D protects against cancer because it is a biological antioxidant. This one is also incorrect. While vitamin D does have some regulatory roles in cell growth and immune function, its main role is not as an antioxidant. That role belongs to vitamin E, not D. So this is also false. Statement 3 says vitamin K is necessary for the post-translational introduction of calcium binding sites. This is true. Vitamin K is involved in the carboxylation of glutamic acid residues on certain proteins, which is going to allow those proteins to bind to calcium. This is really important in blood clotting and bone metabolism. So this statement is true. Statement 4 says vitamin A is metabolized to retinol, which is important for sight. This is also correct. Vitamin A is converted to retinol. This is a molecule that plays a central role in the visual cycle. So we have 2 true statements out of 4, statements 3 and 4, which makes the correct answer for problem 7, C. Let's move on to problem 8. Problem 8 says, which of the following is true of amphipathic molecules? A says they form protective spheres in any solvent with hydrophobic molecules on the interior and hydrophilic molecules on the exterior. Now, this is partly true, but not entirely accurate. Amphipathic molecules like phospholipids or soaps, they can form spheres, but only in aqueous polar solvents. The hydrophobic tails are going to go inward and the hydrophilic heads are going to face the water. But no, this doesn't occur in any solvent. That part is what makes this answer choice incorrect. B says they have 2 fatty acid chains and a polar head group. Now, this is true of phospholipids, but not all amphipathic molecules. For example, bile salts and some detergents are amphipathic, but they don't follow this specific structure. So while this describes a type of amphipathic molecule, it's not universally true of all. So B is incorrect as well. C says they're important to the formation of the phospholipid bilayer and soap bubbles. This is correct. Amphipathic molecules are crucial for forming structures like the cell membrane, where the phospholipid bilayer forms with tails inward and heads outward. And they also form micelles and bubbles like in soaps. So this option correctly captures their function, and it is in fact the right answer for problem 8. We're still going to go through D. D says they have glycerol base. This is only true for specific classes like glycerophospholipids or triacylglycerols, but it is not a defining trait of all amphipathic molecules. So this is not an accurate statement. Again, the best answer choice for problem 8 is C. Problem 9 says, which of the following are true about sphingolipids? Statement 1 says they are all phospholipids. This is false. While some sphingolipids, like sphingomyelins, are phospholipids because they contain a phosphate group, many others, like glycosphingolipids, they do not. These instead have sugar residues and they are not classified as phospholipids. So statement 1 is false. Statement 2 says they all contain a sphingosine backbone. This is also false. While most sphingolipids are based on a sphingosine backbone, some contain variations or derivatives that are sphingosine-like backbones, but not exactly that. So this statement as written is overly strict and technically inaccurate. Statement 3 says they can have either phosphodiester or glycosidic linkages to their polar head groups. This one is true. Some sphingolipids, like sphingomyelins, contain a phosphate group and they form phosphodiester linkages. Others, like cerebrocytes or gangliocytes, they contain sugar groups and they form glycosidic linkages. So this is accurately describing the diversity in head group attachments among sphingolipids. So only statement 3 is true, which makes the correct answer for problem 9, answer choice B. Problem 10 says, which of the following statements about saturation is false? We're going to work through each statement to identify which one is false. A says it can describe the number of double or triple bonds in a fatty acid tail. This is true. Saturation refers to how saturated the fatty acid chain is with hydrogen atoms. If it has no double or triple bonds, it's going to be fully saturated. But the presence of double or triple bonds makes the fatty acid unsaturated. So this statement is accurate and true. B says it determines at least one of the properties of membranes. This is also true. The degree of saturation in fatty acid tails affects membrane fluidity. So more unsaturated tails lead to more fluid membranes, while saturated tails pack tightly and make membranes more rigid. So this is a true statement. Then C says more saturated fatty acids make for a more fluid solution. This is the false statement. Saturated fatty acids are straight and they pack closely together. That leads to less fluidity, especially in membranes. It is actually unsaturated fatty acids, which are going to contain kinks due to double bonds that are going to disrupt tight packing and therefore increase fluidity. So this contradicts the correct understanding of membrane dynamics. It is why it's the false statement and the correct answer for problem 10. We're still going to work through D. D says fully saturated fatty acids have only single bonds. This is true. A fully saturated fatty acid contains only single bonds between carbon atoms in the tail. And this is the very definition of saturation in this context. So once again, the correct answer to 10 is C. Problem 11 says, which of the following is true about glycerophospholipids? Again, here we're going to work through each answer choice. A says glycerophospholipids can sometimes be sphingolipids depending on the bonds in their head groups. This is false. Glycerophospholipids and sphingolipids are different classes of phospholipids based on their backbone. Glycerophospholipids have a glycerol backbone, while sphingolipids have a sphingosine or sphingosine-like backbone. The head group doesn't turn one into the other. So this is not what we're looking for. B says glycerophospholipids are merely a subset of phospholipids. This is true. Glycerophospholipids are one of the two main classes of phospholipids, the other being sphingospholipids. What defines glycerophospholipids is their glycerol backbone and their phosphate-containing polar head group. B is in fact the correct answer for problem 11, but we're still going to work through C and D. C says glycerophospholipids are used in the ABO blood typing system. This is false. The ABO blood group antigens are actually glycolipids, specifically glycosphingolipids, not glycerophospholipids. So C is incorrect. D says glycerophospholipids have one glycerol, one polar head group, and one fatty acid tail. This is false because glycerophospholipids have two fatty acid tails. So once again, 11 is B. Let's move on to problem 12. Problem 12 says, which of the following statements about terpenes is false? A says terpenes are strongly scented molecules that sometimes serve protective functions. This is true. Terpenes are known for their strong aromas, and they do play protective and ecological roles. B says terpenes are steroid precursors. This is also true. Terpenes are the precursors to cholesterol, which is in turn the backbone for steroid hormones. So steroid biosynthesis begins with terpene-based molecules. C says a triterpene is made of 3 isoprene units and therefore has 15 carbons. This is false. Each isoprene unit has 5 carbons. So a triterpene has 6 isoprenes and a total of 30 carbons, not 15. Therefore, this is the false statement we're looking for. 12 is C. We're still going to work through D, however. D says terpenes are made by plants and insects. This is true. While plants are the most common source of terpenes, some insects, like butterflies and ants, also produce terpenes for signaling or defense. So again, C is the false statement because it misrepresents the carbon count of triterpene. Let's move on to problem 13. 13 asks, which of the following is true about cholesterol? All right, let's break this down. A says cholesterol always increases membrane fluidity in cells. This is false. Cholesterol's effect on fluidity depends on temperature. At high temperatures, it stabilizes the membrane and decreases fluidity. At low temperatures, it prevents tight packing and increases fluidity. So it is temperature dependent. B says cholesterol is a steroid hormone precursor. This is true. Cholesterol is the starting molecule for the synthesis of all steroid hormones, including things like cortisol, estrogen, and testosterone. And in fact, B is the correct answer for 13, but we're still going to work through C and D. C says cholesterol is a precursor for vitamin A, which is produced in the skin. Cholesterol is a precursor for vitamin D, not A, so this is false. D says cholesterol interacts only with the hydrophobic tails of phospholipids. This is also false. While cholesterol embeds itself among the hydrophobic tails, its hydroxyl group interacts with the polar head groups of phospholipids. So it spans both regions of the bilayer. So once again, the only true statement about cholesterol here is answer choice B. Problem 14 says, which of the following statements regarding prostaglandins is false? A says, prostaglandins regulate the synthesis of cyclic AMP. This is true. Prostaglandins can activate G protein coupled receptors that lead to changes in cyclic AMP levels inside cells. B says, prostaglandin synthesis is inhibited by nonsteroidal anti-inflammatory drugs, and this statement is also true. Nonsteroidal anti-inflammatory drugs like aspirin and ibuprofen, they inhibit an enzyme that's required for prostaglandin synthesis. C says, prostaglandins affect pain, inflammation, and smooth muscle function. This is also true. This is a hallmark of prostaglandin action. They mediate pain, inflammation, fever, and they contract or relax smooth muscle depending on the tissue and receptor subtype. Finally, D says prostaglandins are endocrine hormones like steroid hormones. This is false, and this is the correct answer for problem 14. And this is because prostaglandins are not endocrine hormones. Instead, they are paracrine and autocrine signaling molecules, meaning they act locally near the site of synthesis rather than traveling through the bloodstream to distant organs like true endocrine hormones. So the false statement here is D. Problem 15 says, which of the statements regarding waxes is false? A says waxes generally have melting points above room temperature. This is true. Waxes are typically composed of long-chain fatty acids and long-chain alcohols, which makes them solid at room temperature. Therefore, their melting points are generally well above room temperature. B says waxes are produced only in plants and insects and therefore must be consumed by humans. This is false. While many waxes do come from plants and insects, animals, including humans, also produce waxes. Therefore, it's incorrect to say waxes are only produced in plants and insects or that they must be consumed for animals. So this is in fact the false statement we're looking for. 15 is B, but we're still going to work through C and D. C says waxes protect against dehydration and parasites. This is true. Waxes often serve as protective barriers, especially in plants where they coat leaves to reduce water loss. and in insects to prevent desiccation and deter pathogens or parasites. D says waxes are esters of long-chain fatty acids and long-chain alcohols. This is true. Structurally, waxes are formed by esterification of long-chain fatty acids with long-chain alcohols, which contributes to their hydrophobic and protective properties. So once again, the final answer for problem 15 is B. And with that, we've completed our practice problem set. Please let me know if you have any questions, comments, concerns down below. Other than that, good luck, happy studying, and have a beautiful, beautiful day, future doctors.